Welcome back to Kenya Fermenter, whereby today we are going to reflect on fermentation. Fermentation was first described by Louis Pasteur as food spoilage, and fermentation is derived from a Latin word which means to boil. So by definition, fermentation is the generation of energy by catabolism of compounds. Biochemists define fermentation as the energy generation process which organic compounds act as both the electron donor and terminal electron acceptance. Fermentation has a different meaning when it comes to the biochemist and industrial microbiologist. So its biochemical meaning is the generation of energy by catabolism of compounds, whereas industrial microbiologists has a broader perspective about fermentation. In fermentation process, microorganisms are used to facilitate the process and catalyze the process of fermentation. Because microorganisms are small and they have a larger surface area to volume ratio, rapid transport of nutrient into the cell, and cell can grow very fast because they possess a wider range of enzymatic capabilities. For example, fungi are saprophyte, which can live on dead, decaying matter. To that factor, we say that microbial reactions are carried out under mild environmental conditions. Most of the microorganisms can survive on low temperature and pressures. While number 4 is that microorganisms are used in the process of fermentation because they can grow plentifully on waste products such as waste pulp, paper pulp, petroleum products, and molasses. Molasses is the waste product derived from sugar cane. There are some products obtained from this fermentation process. What can be obtained from the fermentation process? The end result, the products of fermentations are divided into 5 major categories. Produce microbial cells, produce microbial enzymes, produce microbial metabolites, produce recombinant products, modifies the compound which is added to the fermentation transformation process. The first one, microbial biomass, by fermentation can be used to produce other important microorganisms that include the yeast which is used in baking and those that are used as animal and human feed as a single cell protein. Number 2 is the microbial enzyme. By the process of fermentation, microbial enzymes are generated, and these microbial enzymes facilitate other processes such as enzymes can either facilitate or inhibit a process. So, like for instance, enzyme 1 can enhance the formation of product B, but B cannot be converted into C because of enzyme 2. So, enzyme 1 catalyzes conversion of A into the desired product P, but product B cannot be converted further in presence of enzymes 2. Now, number 3 is the microbial metabolites. Are just the end products of the catabolisms of compounds. And here we have the primary metabolites and secondary metabolites. Primary metabolites are those that are produced during growth process. We know that on a growth process of a bacteria or microorganism, it is divided into 4 phases. That is the lag phase, which there is no increase in the number of the living bacteria. And there is a log phase, which there is exponential increase in the number of living bacteria. And number 3 is the stationary phase or the plateau phase, which there is a constant growth rate of bacteria cells. And finally, the 4th one is the death rate, which there is exponential decrease in the living bacteria cells. Secondary metabolites are those that do not accumulate in the medium until after the cell has stopped growing. Primary metabolites have a close on an arrow range. The red line represents the growth of microorganisms, while the blue line represents the primary metabolites produced. Secondary metabolites, which are the blue line also, do not accumulate in the media until after the cell has stopped growing. So there's a space between, there's a big space between the growth sigmoid curve. L amino acids could be the products or the byproducts of these secondary metabolites. Some examples of primary metabolites include the ethanol, citric acid, glutamic acid, lysine, nucleotides, phenol. We move to recombinant DNA technology. This involves genes of higher organisms incorporated into the microbial cells and are capable of producing heterogeneous protein. E. coli, Saccharomyces cerevisiae, and Fermentations fungi are examples of hosts which can hold these genes of higher organism. Now, from this DNA recombinant technology, there are products which are produced, and these products include the interferon, insulin, human serum albumin, factor 8 and 9, epidermal growth factor, CAF, chymoids. Now, on the entire process of DNA recombinant technology, we are going to design and keep in mind on the secretion product, the end product secreted, while we are minimizing the degradation of this product which is produced, and featuring also on the onset of the synthesis to make sure that we are maximizing the expression of the foreign gene. So to keep in mind is that it is important to consider these factors before undertaking the DNA recombinant technology process. The recombinant technology is expressed so much from gene cloning, which gene cloning mainly occurs in the agricultural sector as in combination of agriculture and technology. We know that basically genes controls the biological molecules, and these molecules include the hormone, enzymes, and cytokines. Now, these genes, when inserted into a bacteria cell, they can grow. And gene cloning involves the desired taking the human desired gene in combination with a small bacteria DNA plasmid factor in presence of DNA ligase. DNA ligase is the enzymes which facilitate training of the complementary DNA gene from human and that of bacteria plasmid factor. Let's consider about the human DNA, which is first cleaned and then it is cut into small pieces using an ECOR1 enzyme, which is called a restriction enzyme. The ECOR1 enzyme is able to recognize specific base sequence on DNA and results in cutting the sticky ends of the double-strand DNA. Now, number two, we need to understand about the plasmid factor and say that these plasmid factor are small circular pieces of double-strand DNA which have capability of replicating autonomously within the bacteria cell. Now, plasmid factor contains small genes that are able to resist antibiotics, such as ampicillin. Host bacterium is that bacterium at which, when the plasmid factor and the desired gene are combined, is placed for growth. It will allow the plasmid factor and the gene to grow. And there are two processes by which the plasmid factor and the gene are inserted into the host bacterium. That is the transduction and electroporation. Before taking combination of the gene and the plasmid factor, we need to test and identify and pick the best quality before transferring them to the host bacterium. Now, this is done by taking a plate of agar, which agar is the culture media, and on that plate we add ampicillin. And on this, ampicillin is an antibiotic which kills the growth of bacteria or inhibits the growth of bacteria. Now, we are going, the benefit of this is to make sure that the other bacteria are killed and the desired combination of the gene and the plasmid factor are the only products that are transferred to the host bacteria. So ampicillin acts on killing the other bacteria and the undesired qualities of the bacterium. So you can plasmid factor contains a resistant gene which will enable it to grow even in the presence of ampicillin. And from this, we are going to do the screening. On this process of selection, screening is done so as to identify the best combination that can be transferred into the host bacterium. Now, this screening is done to identify the best, and what is selected is a white colony. The plasmid factor is designed in a manner that it contains a restriction site with a beta galactosidase gene, which can either be intact or defective. And due to this beta galactosidase gene, foreign colonies will produce white colonies which are easily picked and transferred to the host bacteria. So that is how the process of selection and screening is done. Now we go to the transformation of compound as the product produced by the process of fermentation. Microbial cells can be used to convert a compound into structurally related or financially more valuable compound. We say that this process involves the use of bacteria to convert these compounds into heterogeneous proteins. The benefit of using the microorganisms is that these microorganisms can be reused as many times as possible as catalysts. They only catalyze conversion of the compounds into structurally more valuable products. Because the microorganisms can behave as a chiral catalyst, and these processes in which the microorganisms are used as a chiral catalyst include dehydrogenation, oxidation, hydroxylation, dehydrogenation, condensation, carboxylation, amination, deamination, and isomerization. Microorganisms are able to transform compounds. For instance, steroids are naturally occurring in the body. For example, adrenal corticosteroids, which contains the cortisol and the corticosterone. Now, prostaglandin hormone contains the progesterone. Androgens contain the testosterone. Estrogens produce the estradiol and estrone. In this process of transformation of compounds, products such as anti-inflammatory drugs, sedative, anti-tumor drugs, cardiovascular drugs, oral contraceptive, and dermatological agents have been produced. Here are examples of enzymes, proteins, and polysaccharides produced by microorganisms for commercial purposes, and there is a range of pharmaceutical formulation. Now, remember, we've begun by defining the term fermentation as the process of catabolism of compounds, mainly carbohydrates. Regardless of the types of fermentation, the following 6 steps are necessary. The first one is you formulate the media to be used in culturing the microorganism. Now, number 2, we do the sterilization of the media, fermenter, and the equipments. Number 3, we produce the active, pure, and sufficient quantity of the culture that is able to inoculate the production phase. Number 4, we grow the organism in the production fermenter under optimum conditions. Number 5, we extract the product and perform the purification process. And finally, we dispose of the effluent produced by the process. Now, we are going to reflect on the basic function of the fermenter. The basic function of a fermenter is to provide control environment for the growth of microorganisms in order to obtain the desired production. Now, there are some consideration points to make and to put in place in designing and constructing of the fermenter. Now, the vessel should be capable of being operated aseptically. Number 2, it should have adequate aeration and ease of agitation. Number 3, power consumption of this fermenter should be low. A fermenter should have a temperature control system, should have also a pH control system, should be able to provide sampling facilities. A fermenter should have low evaporation losses. A fermenter should be designed in a manner that it uses less labor in operation, harvesting, and cleaning and maintenance. A fermenter should be suitable for a range of the processes and should ensure smooth internal surface. And cheapest material should be used so as to ensure the cost is effective. Now we have types of fermenters. Types of fermenters include the batch fermentation. We have types of fermentation, which includes the batch fermentation, continuous fermentation, fed batch fermentation, and anaerobic fermentation, aerobic fermentation, surface fermentation, submerged fermentation, solid state fermentation. The anaerobic fermentation takes place in absence of oxygen, while aerobic takes place in presence of oxygen. But there is this batch fermentation and continuous fermentation. Batch fermentation involves the stopping of the fermentation process while removing the required so as to withdraw the products, while continuous involves that which the process of fermentation continues regardless of withdrawing the product. So you withdraw continually and you produce continually. So we have two types of fermenters, and these types of fermenters include the batch culture fermenter, where the microbial growth is altered to allow for the removal of the required product. Whereas in the continuous fermenter, the continuous culture is where the microbial growth is maintained at exponential level and the product is continually removed. So the process continues and the removal also of the products continue. Now we are reflecting on the commercial benefits and commercial products of fermentation. Remember, fermentation was first defined as food spoilage by pasture, but fermentation over time has transformed itself to more benefit process, and it is commercially used to produce various range of the products. Now, these products are derived mainly from the process of biotechnology, where these microorganisms are used in production of many important products, and these commercially important products include the antibiotics, amino acids, organic acids, enzymes, vitamins, biosurfactants, catalysts, biopolymers. In the productions of antibiotics, such as penicillin, fermentation process is involved to control precisely the nutrients. And on biocatalyst, the transformation or biotransformation by the help of microorganisms, biotransformation to modify a steroid is possible. Microbes are products of fermentation. Commercially, also the microbes are important, for instance, in the marine biosensor design. Let's look at the production of steroids today. Steroids are produced from plant steroids. Stigmosteroid undergo the chemical steps to produce the progesterone, and the microbial steps, and the progesterone undergo microbial steps to produce 11-hydroxyprogesterone, and can either go through a chemical and microbial step to produce prednisolone, or can undergo chemical steps to react with compound S produced by dioxolin, and in the end, hydrocortisone produces the cortisone, and finally the prednisone. So this is just an illustration. And finally, the other importance or application of fermentation is this production of other chemicals, which are the biologicals. These biologicals is hormone, vaccines, and etc. Now we say that fermentation is extensively used nowadays in the production of antibiotics. And fermentation process has been applied mainly in the production of penicillin, and specifically penicillin G. Now, penicillin was first discovered by Fleming and described Penicillium notatum. Over the period, developments has occurred on the strain selection, which can produce higher yield. So, for instance, that which Fleming used was Penicillium notatum, which could produce 0.3 microgram per liter. And over the ages, there is continuous improvement on the penicillin producing strains through the mutation and selection, which the current strains produce 30 grams per liter. To explain on the production of penicillin by feedback method, where the first penicillin was purified by Florey and chain in 1940. Now, the first products was aerobically submerged fermentation using penicillium chrysogenum. Penicillin is a secondary metabolite, so that is only produced on stationary phase. That is ideal phase. Now, penicillin being a secondary metabolite requires only a stationary phase, and this stationary phase is obtained only by batch fermentation and fed batch processes. Now, because they prolong the stationary phase and thus increase the production. Now, penicillin production by fed batch method on the upstream process, where upstream fermentation process involves sterilization of the medium, fermenter, inoculum preparation, and under controlled environment production. Now, this one, upstream involves the regulated pH, temperature, aeration, and agitation. For constant liquor is added as a nitrogen source as well as the phenyl acetic acid to produce penicillin G, with manganese, copper, iron, zinc as trace element for vitamin B12. To the sterile media in the fermenter, culture is added on and left for 6 days. So that is just an illustration, a brief summary. From the upstream process, 30% of the culture is removed and 30% of the medium or fresh medium is added. Now this is to prevent overflow and to facilitate agitation and aeration. Now this is an example of fed batch process and is partially closed system. So the temperature ranges from 25 to 28, pH is 6.5, and the forming agent is the vegetable oil. Now downstream process. Now as penicillin is secreted into the medium after production, the filtrate from the fermenter is then filtered through a rotary filter and all fungal cells are removed. So on this, sulfuric acid is added to lower the pH to 2, at which penicillin is soluble in butyl acetate. Now, extraction proceeds by countercurrent extraction. That is the method at which penicillin. This is done at low temperature because penicillin is stable at acidic pH. Now, to the filtrate, again, phosphate buffer is added to raise the pH to 6.5, and penicillin is extracted into water by countercurrent extractor. The process of acidification and extraction is repeated, and finally, water-soluble penicillin is extracted as a form of potassium salt, by reacting with potassium hydrogen. Hydrogen salt is precipitated, washed, filtered, and dried to get 99.5% pure penicillin. The final product of industrial fermentation is benzyl penicillin, which is the penicillin G. Now, penicillin G is limited to gram-positive. It is acid labile, meaning it can be destroyed easily by the gastric acid in the stomach. Chemical analysis of broths always give a higher concentration than the biological analysis. Now, it's important to know of the 6-aminopenicillic acid, which is just used as a starter material for making penicillin. Summary of the fermentation process. You first take the penicillium chrysogenum spores, and secondly, you place the penicillium chrysogenum spores into a 500 ml flask containing 100 ml culture, and you leave it for 4 days, and the temperature should be maintained at 25 degrees Celsius. And the third step is you transfer the content into a vessel of 2 liters. And on this, let it to stand for 2 days. Number 4, you transfer the content into a septic tank, 500 liters, and you let it to stand for 3 days at 25 degrees Celsius also. Thereafter, you transfer the content in the seed tank into the fermentation tank, which is about 180,000 liters for 5 to 6 days at 25 degrees Celsius, remember? So from there, you pump broth through the rotary filters. After pumping through the rotary filters, you extract the amyl acetate. Then you transfer the phosphate buffer, and you will obtain crystals by the process of crystallization. Then you wash the crystals. Thereafter, you do the filtration again to remain with crystal and to ensure that you are obtaining clean and purified. Thereafter, you wash. After washing, you filter. After filtering, you take it to a vacuum dryer, and finally you will obtain crystals of crystalline penicillin G. That is the summary. Thank you for watching this discussion about the fermentation process and its application, especially in the production of penicillin. And I hereby ask you to subscribe and share so as to get more notifications on our new videos.